Well, good afternoon, everyone. My name is Tricia Johnson. I'm a part of the Learn America team that has been preparing a natural biology review to help you succeed in the nursing program. You have each received the results from the biology portion of the recent diagnostic tests you took where your strengths and weaknesses were identified by the topic. This is the first of our 4 two-hour lectures that will be presented. The topics will help to improve your biology skills, and this will help to strengthen those skills. During the 2 weeks following the lecture, there will be 4 one-hour sessions. During this time, you will have the opportunity to ask any questions you would like about those topics. I'll cover, or any questions covered during the lectures, and the problems that you'll work on, we could go over them to check your understanding of that section. Remember to please consult the review schedule that has been sent to you for all upcoming lectures and online help sessions. We look forward to being a part of your success at LIU. Good luck, and now let's get started. Right now, I want to start from the basic structure of biology. So we're going to go back to where we start with the cell itself, and then we're going to work from there and talk about each component or each part of the cell. We'll move from the cell and look at all the organelles that make up the cell, how those structures actually work together to carry out homeostasis in the cell. Once we've done that, then we'll take it from the cell and look at how the cells come together to form tissue. The tissue in turn will form the organs, and organs develop the organ system. So we're going to start from the simplest component of the human structure and take this over the course of the 8 weeks into the development of the human body systems. So the first section, I like to call this Journey Through the Cell. Now we see the biology is the study of living things. We are going to study also some of the abiotic factors that affect the biotic factors, which are the living things in our environment. Now, the characteristics of any living thing will definitely show that it has to have a definite form. It has limited size. Anything that's considered to be living is made up of cell, as cell is the basic unit of all living thing. Living things contain some sort of genetic code. It's universal for all living things. It's acquired also that living things process nutrients and be able to use those for energy. Living things are also made of complex chemical compounds. They have a finite lifespan. They're able to grow and reproduce, even though reproduction is not needed for survival of the species. It is part of the life function needed by living things. All living things need to be able to respond to its environment. So one of the first living things, life process we would like to look at is nutrition. And so the process of taking in or obtaining materials from your environment, the organism, which is another term for living things, has to be able to take in the raw material and process it. And what's meant by processing the raw material? We're talking about being able to digest, break it down, or convert it into a form that can be used. And so we'll see that different living things are able to process materials in different ways, but it doesn't matter the species, it has to be able to process it into a form that it could be used. Respiration is one of our more detailed life processes that we'll cover in more extent as we move through the 4 weeks, the 4 lecture sessions. Respiration, a lot of people think respiration is just breathing, processing air in and out of the body. Breathing is one of the processes that aid in respiration. That's how you'll get the gases that's needed for converting food into the form of energy. So when you see respiration, now you need to think of energy. It is the body's way of converting food into energy. Let's see here, let me just check on my battery power here. Sorry, accidentally. All right, so like my computer, our body needs energy. And respiration, whenever you come across respiration, you should automatically think of energy. Transport is the other life process that we're going to cover extensively. And transport, whenever you think of transport, we're talking about key words to look for when we are referencing the different life processes. And one of the key words to look for is material circulating through the organism, materials moving in and out of the organism. So circulation, transport is the movement or circulating of materials within and out of the cell throughout the body. Locomotion is different, as in locomotion focus on the actual movement of the organism from one area to the next, whereas transport is specifically related to diffusion of materials going into and out of the cell. So we want to differentiate between the two of transport and locomotion. Growth. Growth is one of the life processes that's very important to a living thing. And say growth doesn't have to be necessarily in height, doesn't necessarily have to be in mass, but different. It could be in volume, it could be expressed in different ways. So growth has to deal with the size of the cell itself. Because if you think of the organism, initially begins with one cell, and that one cell will then divide and increase to become more than one cell. And so it's this process that we're referring to growth as, and not just an increase in height. We reference height mostly when we look at maybe animal, we're thinking about mammals, but a single cell organism is only made up of one cell. It's unicellular. So we're looking at the fact that there could be an increase in volume, a change in the cell itself. So growth, increase in size. It also, when we're talking about simple organism, it could be an increase in volume or surface area. And even as we think of ourselves, we initially started out with one cell. And through growth and the process of mitosis, that one cell was then able to develop and divide into many cells to the point where there's an increase in surface area. Reproduction, as I mentioned before, is characterized as one of our life processes, but it's not necessarily needed for an organism to maintain life. And reproduction itself is where a species will make one of its own kind. It will reproduce that specific species. There's two ways that this process can occur. It could be through sexual reproduction or asexual reproduction. And we will be exploring these two concepts. And so in sexual reproduction, we'll see where we'll need a male and a female gamete in order to make the new species. Whereas in asexual reproduction, we have a variety of options or styles of reproduction, whether it's through budding, as you could see here on the organism itself. You could see there's a budding starting here, and in this section here, you could see the daughter cell being produced. So that's asexual reproduction. Next, metabolism. Metabolism referred to all the chemical activity that's occurring in that organism. So we're referring to all the cellular process. We're going to see enzymes involved in this process. Anything that's related to chemical process taking place in the body. And so we see that there's an anabolic and catabolic processes that take place. So if we look at synthesis, your body will use simple compounds that's taken in to create or form more complex organism. And so this process, we'll see its development in the muscle cells or creating different parts of the cell. Where catabolic gets a little bit more detail, but it's the removal of any of the metabolic waste from the cell.
In this video, you will learn about the cell. Bricks, which are used in construction, are the basic structural units of buildings. Similarly, cells are the basic structural units of living organisms. Buildings are constructed in different shapes, sizes, and designs, but the bricks used are made up of the same materials and are similar to one another. Similarly, cells are complex living structures and often are studied using a microscope. From this video, you have learned about the cell.
And so I just wanted to give you a quick intro into the cell based on that video, just to show you how the cell itself, once again, is the basic structure. We will also be discussing the microscope and how that the development of the microscope gave us the opportunity to explore the cell and learn more about the cell itself. Now, cells are characterized into two categories. They're prokaryotic cells and they're eukaryotic cells. And once again, as we move through today's lecture, we'll discuss them both in greater details. The prokaryotic cell will be more of your bacteria cells. These guys have no membrane-bound structures on the inside. So structures such as the nucleus, the mitochondria, Golgi body, chloroplast, these structures you do not see wrapped in the membrane inside the cell. Whereas the eukaryotic cells, those have the true nucleus and the organelles. And so that's the one we refer to the most when we talk about plant and animal cells. Those are our eukaryotic cells. Whereas the prokaryote, as I said before, are primarily bacteria cells. I want to take some time to just review the microscope, because understanding the microscope and understanding how to use the microscope, once you have this skill, develop this skill, it's going to enhance your ability to observe the cell properly, and it will give you more confidence on what you're seeing. And by understanding the cell, that will give you more confidence in the concepts related to the cell. So here we have, if you take a look, we'll start at the top. Just a quick review of the eyepiece. We'll start out at the top. So the eyepiece itself, pointed here, usually has a magnification of 10. And the eyepiece has a lens in it. And that's where you're getting the magnification from. The body tube is this area here, the tube down into this section. So the body tube creates a distance between the eyepiece and the objective lens. And if you take a look at the objective lens, there are different lenses. It depends on the one that you have available to you in your lab sessions. They have varying magnification. And so the distance between the two allow you to see the image better. And so whatever you see in the microscope is reversed and backwards. So if I was to be looking at the letter F, it would look like that naturally, but under the microscope, it would be reversed and backwards. And so by keeping that in mind, when you look under the microscope, that whatever you're seeing will be reversed and backwards. Also, when you move the object under the microscope, if you need to move it forward away from you, you'll move the slide towards you. So it's going to be opposite all of your motions. If you would like to move the object to the right, you have to If you would like to see the object to the right, you would have to actually physically move the slide to the left and vice versa. So everything that you see is a reverse reflection. The stage here is where you would put the object and you will fix them in place by the clips. The diaphragm is very important, and the condenser is mentioned in this image. The diaphragm is where you would use to control the lighting that's going to pass through the floor, pass through the stage, and send the light up into the objective lens so you could get a clearer image of the object. So if you're looking at an object that is unstained, you will probably lower the light intensity so that you can see more details. If it's a dark substance, something that's heavily stained, you're gonna want to brighten the light by using the diaphragm. So the diaphragm usually has different size openings, and there's diaphragm and disc diaphragm. And so that allows the light, which is down here, to pass through the specimen up into the objective lens to allow you to see the object. Very important objects here on the side. You have your coarse adjustment knob and your fine adjustment knob. So the coarse adjustment knob is if you want to move the specimen closer to you or away from you. You would use the coarse adjustment knob. And if you wanted like a greater focus, use the coarse adjustment knob. If the object is still, I would say, foggy or not clear enough, that's when you use the fine adjustment. It gives you more detail or more depth to the image when you use the fine adjustment knob. And so the fine adjustment knob, it's not suggested that you use the fine adjustment knob when you are under the high power objective. A possibility, if you leave the high power objective on and you're using the coarse adjustment, you don't want to use that under the high power objective. If you use that, you can crush the slide itself. So let me clear that up. When you're using a high power objective, you want to use the fine coarse adjustment, not the coarse adjustment knob. Okay, and so in this particular one, you have your base and your light source at the bottom. So it's very important that you understand how to use the microscope when you're, especially your microbiology studies. Having a strong skill in the scope itself will definitely increase your confidence in what you're seeing and knowing what you're supposed to see. All right, so let's take a second and look at the prokaryotic cell. So prokaryotic cells are referred to as bacteria cells. So bacteria is referred to as the first living things to be found on the earth. Their characteristics are different than the cells that evolved after bacteria cells. So if you take a look at the image here, we have different parts to the cell itself. So if you take a look inside, I'm going to point inside. Here you have your DNA structure. It's referred to as a nucleotide. In this image, it's the red structure. So what you see is that the DNA is just floating throughout the cell, and it's referred to as a nucleotide. You still have a cell wall, you still have a cell membrane around the structure itself, but what's missing is that nuclear membrane to hold the DNA in a circle. So what you'll just see is the DNA floating around, and it's referred to as a nucleotide. We still see a cytoplasm here. We see the flagella. The flagella is this tail-like structure here that's being shown. And the pili, very important structure for bacteria reproduction. So we'll see that play a role whenever material is going to be passed from one bacteria to the next. And that's one form of reproduction for prokaryotic cells. We have plasmid here, which we're going to discuss later when we get to our genetic unit. The plasmid is a ring of DNA that in our culture today, we use that for genetic engineering. It's extracted biotechnology where they're able to cut pieces of the DNA and insert new information in. So we will also spend some time discussing that in greater details. And so, as I had mentioned before, the plasmid, it's a circular DNA. It's present in all bacteria. And this material has been used, once again, growth hormone. We see a lot of hormones made from this process of genetic engineering using the plasmid. The flagellum in itself is how the bacteria will get around and move through locomotion. It's a tail-like structure. And as I've mentioned before, the pili is not necessarily for movement, but it's an appendages for reproduction. Okay, so let's move on to the next category of cells. These are the eukaryotic cells and the ones that we're most familiar with. And so these eukaryotic cells, we have the plant cell and the animal cell. I believe most of us are more familiar with these structures. If you take a look, we have all the nucleus we find in common in both cells. We could go around. Let's start over here in the plant cell. We could see the cell wall. So around the lining of the cell is where you have the cell wall. The cell wall, the inner wall on the inside of the cell, if I could just keep my hand sturdy here, on the inside, that's where you're going to find your cell membrane. But the cell membrane is also referred to as the plasma membrane. So if you see plasma membrane, it's referencing the same thing as the cell membrane. It's referring to it's the membrane around the cytoplasm. The other structure that we'll review is the endoplasmic reticulum, sometimes referred to as the ER. Ribosomes we're going to find throughout the cell and bound to some of the structures inside the cell. We have vacuoles, and as we could see in the plant, we have a large vacuole here, whereas over in the animal cell, the vacuole is much smaller than in the plant cell. Chloroplasts, we're going to see the green-like structures over here, and we'll review those because a lot of the structures, we have similar structures in plants and animals. And we have the cytoskeletal material in the plant cell. That's the stuff that help in the structure of the cell. We also will touch on the mitochondria as providing the energy for the cell, the Golgi apparatus, cytoplasm, and the centriole. So we're going to go through, take some time to review some of the major organelles found in each one of these. You just check to see here if we're ready to go. All right, yep. Let's take a look at the size of the cell. Let's watch this video.
Cells are the basic building blocks of living organisms. Their size and shapes appear in many different lengths. A small molecule that builds the cell measures only about 1 nanometer.
And guys, let me just interrupt here. So I want you to take note of the measurements on the bottom, where you have centimeters on this side, and here's millimeters. Remember, we have 10 millimeters make up 1 centimeter. So the size, we're looking smaller than that. Where we have 1,000, that U that's there referred to micrometers. Then we have nanometers. So we're talking about something that's smaller, 1,000 times smaller than a millimeter. So that's how small we're looking at these compounds.
Cells are the basic building blocks of living organisms. Their size and shapes appear in many different lengths. A small molecule that builds the cell measures only about 1 nanometer. The size of a virus ranges from 10 to 100 nanometers. There are several bacteria whose sizes range from 100 nanometers to 1 micrometer. Animal cell sizes extend up to 100 micrometers, and a plant cell can be as large as 1 millimeter.
Okay, so just a quick reference or referring to the size of the cell, just to give you an idea of how small it is. If you were to take 1 millimeter off a ruler and break that 1 millimeter into like 1,000 pieces, one of those small pieces will give you a range of what a plant cell might be. And think about the chemicals inside of that would be even less. All right, so let's go on to our first organelle. And so the first organelle we're looking at is the chloroplast. And we will be talking about the chloroplast in more detail in session 2, lecture 2, when we explore DNA and the genetic compound of an organism. But right now, let's review the basic structure of the nucleus. So here we have the nucleus. We have an image here to your right. The nuclear membrane is also referred to as the nuclear envelope. And so what you could see are there are actual pores in it. So materials are able to move in and out of the nucleus itself. In the center, we have the nucleolus. And we're going to see that this nucleolus will eventually play a role in the production of the messenger RNA when we're talking about transcription of the information for the making of protein in the organism. Floating inside of the matrix of the nucleus, you have the chromosomes. And so the chromosomes are the genetic information that was inherited by that organism. For example, for us human, 46 chromosomes were inherited. We inherited 23 from mom and 23 from dad. And so we have 46 chromosomes. Different species carries different number of chromosomes. The chromatid are string-like structures found floating in the nucleus, and those materials can condense to form chromosomes. So once again, the nucleus, the role of it really is to carry that hereditary information. It contains the DNA, a packaged condensed DNA in the chromosome. So if you condense the DNA, it forms a chromosome. And so it has the genetic information, the instruction that's needed for the cell to function. So every organelle rely on the instruction from the nucleus. So it's referred to as the central, pretty much like the headquarters of your cell. Here's where it's going, here's the organelle that's going to send out that genetic code to each cell to tell it what function needs to be done. The next organelle, I have it separated. So here's a different view of it down here on the left. And so as you look at it, the nucleus we just discussed is right in here. And so attached to the nucleus, floating around the nucleus, are two types of endoplasmic reticulum. We have the rough endoplasmic reticulum, and we have the smooth endoplasmic reticulum. So the rough endoplasmic reticulum, one thing that differentiates the two between each other is that the rough endoplasmic reticulum has ribosomes attached to it. So ribosomes is another organelle that we will go into greater details about, but they're also found on the rough ER, which give them their characteristics. That's why we call them rough endoplasmic reticulum, because the ribosomes are attached. Whereas the smooth endoplasmic reticulum, we don't find the ribosomes attached. So we find ribosomes attached to the rough endoplasmic reticulum, and we also find them floating throughout the cell. So as you could see here in the definition, the smooth endoplasmic reticulum assist in the synthesis of lipids. So we're talking about our fats that are going to play a role in steroid hormones. They, once again, are connected to the rough ER, to the Golgi apparatus, and they're also involved in detoxification. So the rough ER is the site where protein synthesis will occur, because our ribosomes are where protein is made in the cell. And so we're going to see some of that process taking place by the endoplasmic reticulum due to the presence of the rough ER. I wanted to give you guys another chance to review just the structure of the endoplasmic reticulum So let's take another minute and observe this video.
The endoplasmic region of the cytoplasm contains a network of interconnecting membranous vacuoles and cavities. This network is called the endoplasmic reticulum. It is normally associated with the nucleus.
So I just want to step in right here to show you the endoplasmic reticulum. Notice that as it's attached to the nucleus, there are actual channels that move through the endoplasmic reticulum. So it's almost like a pathway. So you're going to see chemical reactions taking place where materials will move in the inner matrix and some will move out of it. So I want you to just see just the layout of it. And we're going to see that there are going to be vessels form off the endoplasmic reticulum. Let's continue.
Nucleus of the cell. The endoplasmic region.
All right. So attach where we saw those vessels will be the Golgi apparatus. This is another structure that's found inside the cell. And so the Golgi apparatus, sometimes referred to as the Golgi complex or the Golgi body, these are different names that are associated with it. Same thing, has that same channel format, but the characteristics that we'll refer to it as are almost like flattened pancakes, like because it's like a stack of these structures on top of each other. And the way you distinguish this in comparison to the ER, the rough ER, is that you will not see ribosomes attached to this structure. And most of the time, if not all the time, you're going to see these vessels floating along the side. You're going to see vessels moving. So because of that, the Golgi apparatus is referred to almost like the post office of the cell, where materials are packaged and distributed throughout the cell. And we'll see that materials such as lipids or even enzymes form from the lysosomes will be packaged and sent throughout the cell. All right, let's take a look at the mitochondria. So the mitochondria is the next organelle that we will spend extensive time on, but as of right now, we just want to cover the basics. So the mitochondria itself, let's take a look at the image. Here has an outer layer, and there's an inner layer to it. And that's referred to as the inner membrane and the outer membrane. Throughout, you have this folded structure on the inside of the mitochondria. And the space that's formed along is referred to as the intermembrane space. In between each fold, that's referred to as a matrix. And you could see again, here we have the ribosomes found throughout the structure. And the mitochondria, the primary purpose of the mitochondria is to manufacture energy for the cell to use. So respiration takes place by way of this organelle. You will see, we will discuss the chemical processes that takes place in the mitochondria. And one of the processes referred to as the Krebs cycle, where sugar is converted into different forms that the cell can use for energy, and the electron chain transport. We'll see that the outer membrane is selectively permeable, meaning that it allows certain substance in and out. So the substance that we'll see is ATP, ADP, and ions and nutrients passing through the structure. ATP is referring to the energy molecule that's made by the mitochondria. ADP is a lower energy molecule. So we're talking about nutrients being converted to energy, and that energy that we're looking for are ATP molecules. The ATP molecules is used throughout the body to provide the muscle cell, provide energy for chemical reactions. And then we can see that that inner membrane is only permeable to water, gases such as oxygen and carbon dioxide. Oxygen is the gas that's needed for aerobic respiration. And we'll see that it will diffuse into the mitochondria to carry out cellular respiration. Carbon dioxide will be one of the byproducts, the waste that will move out of the mitochondria, which eventually will be removed from the body. Another important organelle for us to cover in this session is the lysosome. So the lysosome, we don't hear about the lysosome as much, but the lysosome is very important. The lysosome carries enzymes, and its job, it's there to protect the cells from infection. Any worn out cells, it will be its job to get rid of the worn out cells. It will also aid in breaking down or digesting proteins, fats, different sugar compounds in the body. And also, which I found is important because we always talk about the sperm cell having enzymes, but we don't go into details about it, that it's in the acrosome, which is the enzyme pocket at the top of the sperm cell. The enzyme in it, you have the lysosome is found in that area, which will give the sperm cell the enzyme that it will need to break down the outer layer of the egg cell during the time of just before fertilization. So the sperm cell has to burrow into the egg cell, and it's going to use the enzymes from this lysosome to help break down the outer shell in order for it to enter the ovum or the egg cell. So if you take a look at the diagram here, we're showing that the enzyme materials are found inside of the lysosome, and it has a membrane, because remember, this is eukaryotic cell. It has this membrane which allow materials to move in and out of it. So right now we could take a quick look again. Let's go back to that original image that we saw and look at what are some differences between the plant and animal cell. Because most of the organelles we just covered contains both those organelles. So I'll give you a second just to check it out. Some of the major differences. All right, so here are some of the differences. In a plant cell has a cell wall. The vacuoles are larger, and it contains a chloroplast. We will not find any centrioles in the plant cell. The centrioles are involved in reproduction of the cell. We see these during mitosis and meiosis. Because the plant cell has this rigid cell wall, centrioles are not needed for the fibers to anchor to. In the plant cell, those fibers will anchor to the strong fiber substance in the plant cell. Well, in the animal cell, once again, we do not see a cell wall. The vacuoles are much smaller. We do not carry chloroplast, and we see the presence of the centriole, because the centrioles will serve as the anchor for the spindle fibers that will attach to the chromosome to help in division or making copies of the genetic information in our cells during cell reproduction. All right, let's take another look at that cell wall surrounding the plant cell. And so the cell wall is very rigid. And one of the reasons that it's so rigid, it has cellulose. Cellulose, it ends in that ose, which reminds you that it's a sugar. And so this complex molecule helps to bond like hydrogen bonds together. The sugar molecules help to cause an interlocking connection, and that's one of the things that give it its rigidness. We have pectin, which also will give the cell its porosity, allow materials to move in and out. We have protein and hemicellulose, which is also another sugar. All of these work together to form this interlocking structure to give the cell its rigidity. So in general, the general function of the cell wall is that it's going to surround that plasma membrane. So we saw that the animal cell also have a plasma membrane. The cell wall is there to protect the plasma membrane in a plant cell. So that's what gives the plant cell structure and support. And as I said, we already spoke about the sugar compound that's formed in the cell wall. All right, I want to review some components of the chloroplast. So the chloroplast, it's not found in animal cell. We see it only in photosynthetic organisms and plants. And so the chloroplast, I've pulled an image. So if you take the plant cell, and here let's zoom in to the chloroplast. The chloroplast here is an organelle to itself. Inside of the chloroplast, you have these stacks, which are represented or referred to as a grana. Each stack, or referred to each pancake in this grana, is referred to as a thylakoid. So we're going to see that during the process of photosynthesis, photosynthesis occurs in the chloroplast. So photosynthesis is the process by which autotrophs, these are organisms that are able to produce their own food, such as glucose and sugar. So we're looking at plants, blue-green algae. These organisms, they're able to capture the sunlight, use that sunlight, and convert it into some chemical energy that they could use. Whereas for us, we don't contain a chloroplast. We rely on eating and taking in that nutrients. The photosynthetic organism, photosynthesis takes place in the chloroplast itself. What happens is the sunlight energy will get trapped in the thylakoid. Enzymes in that process will help to split the light molecule. It's called photolysis. And as a result, oxygen will be released, and the hydrogen would end up in the space that we find here around the grana. This space is referred to as the stroma. And it's in this space where we refer to as a dark reaction takes place. That's where the sugar is made in the plant cell. So the thylakoid is where light is split. This is a light-dependent reaction. This is where the oxygen is produced. This is where the hydrogen will then be released to be a part of the dark reaction. We'll see hydrogen carriers come and take that into the dark reaction. So this is just to give you an overlook and insight to the chloroplast. And remember, at the end of this, we're expecting sugar to be made from this process so that the plant can use it for its own respiration process, cellular respiration. So to just give you another view of what I just mentioned, and I have another image here on the side, just to condense that information. So I do suggest you go back and take a closer look at this once we're finished. So the light reaction, this is the part of the reaction that's dependent on sunlight. It's referred to as the photochemical portion of the photosynthetic process. So during photosynthesis, you have your light that enters into the cell. Remember, think to yourself right now, what do we call C? Right, that was our grana. And each one of those stacks referred to as a thylakoid. So the sun's energy is going to get trapped in that thylakoid. Think about what's another thing that plants need. We need light. They also need water. So I'm going to use H2O. Okay, let's try that again. H2O. Water is going to enter in. And so we're going to see during this process, water is going to get split, which is referred to as photolysis. And there are two systems. There's photosystem 1 and 2 that will take place. So we have chemical reactions that will take place in this process here in order for oxygen to be released through the light reaction process. The hydrogen will then flow into the stroma, which is the dark reaction side. And so let's go to that slide and take a look. All right, maybe went too far. It's referred to as the dark reaction or also the light independent reaction. So what happened in this section is that we have the gas entering, which is carbon dioxide, will enter into the plant, into the chloroplast. And so in the stroma, we have the carbon dioxide, we have the hydrogen carriers coming from the process that took place in the light dependent reaction. They're going to get processed through a cycle referred to as the Calvin cycle. Once again, dark reaction, or you may also see it listed as carbon fixation. And so as that chemical process takes place, as a result, the carbon, hydrogen, and oxygen would end up forming that sugar that the plant needs for respiration. And so these two processes are needed to change light energy into a chemical energy. And so here's that image. I really like this image because it's simple, it's straightforward. Where you see here is the balanced formula for photosynthesis, where you have carbon dioxide going in, water, sunlight is there converting those elements into C6H12O6, which is that organic matter, which is sugar, and also the oxygen is released from that light reaction. So as a quick review, A represents water, C is where we're going to have those energy transfer chain taking place, and E will see oxygen being released. B, carbon dioxide will be entering the cycle. Remember, that's the Calvin dark reaction, can also be referred to as carbon fixation. And here we'll see sugar being made. And G is showing you that this entire thing is the chloroplast. All right. So there's other structures in the cell that's important. We have the plasmids and the chemoplasts and the chloroplasts, which we just mentioned. These structures are used to create the color in the plant. So chlorophyll is responsible for that green color that you see in the leaf. So photosynthesis takes place in the green portion of the plant, in the leaf portion of the plant. So the flower of the plant could refer to as the fruit of the plant. All right, so let's take a look at a few questions. Just give you a second to just kind of digest some of the things that we just spoke about. And let me just discard some of our images here. Move some stuff around. And okay, so let me go back. I want to show you here are some questions. So let's process some. Let me give you time to look at it. Let's take a look at structure 1. Structure 1. Structure 1 is pointing, my image. Here we go. So structure 1 is pointing to this dot, that circle on the endoplasmic reticulum. So structure 1, think to yourself, do you remember what that is? So structure 1, should have guessed, was our ribosome. The function of the ribosome, which is our second, the function of the ribosome is there for protein synthesis. And so the life process that's associated with is synthesis, taking simple substance and making something complex. And so we're going to see later that that structure is going to help in producing proteins for the cell and for the organism. Okay, good. Let's take a look at the second one. So it's pointing to this organelle in the middle. I'll give you a second to think about it. All right, so structure 2 represents the nucleus, the function of the nucleus is to control cellular activities. So I know some people probably just say that it contains genetic information. That's fine, but the primary function of it is to control the cellular activities. The live function will be, it'll be involved in distributing genetic information for reproduction. And the cell uses it to maintain homeostasis. Structure 3. Let's take a look at that. Structure 3. So the giveaway for structure 3, when you're looking at these images, structure 3 I'm going to draw on the side here. Anytime you see like folded structure on the inside, is a giveaway that that's the mitochondria. And so in structure 3, we see it being the mitochondria, so the function of it is for cellular respiration. Its life process is respiration, which allows the cell to produce energy for all of its activities. All right, so that was a quick practice question. You can always go back to this and review it for yourself. All right, so now we're going to move on to the membrane-bound structure. This is the cell membrane, and so the cell membrane is crucial to the function of the cell. The cell membrane, once again, is also known as the plasma membrane. It's semi-permeable, which aids in the entry and exit of the substance into the cell. It contains receptors, and mainly, a lot of people forget about this, it's used for communication. So there are receptors that allows materials to move in and out of the cell. Let's look at the components of the cell membrane. So we have the purple section of this image. It's referred to as the phospholipid structure. So you have the top portion, and there are these little dangly portions here. Here's another part, and the dangly portion. They're connected. This section, these are fat lipid structures, which are fats that makes up the cell membrane. This portion here is referred to as being hydrophilic, whereas the inner portion is referred to as hydrophobic. And so the way these are aligned, it's aligned to allow materials to move in, if it's inside the cell, allowed to pass through and go out the cell. And it's also meant for materials to pass out to inside the cell. And so that's why it's referred to as being selectively permeable. Cell membrane, two main components. One is the lipid portion, and then the other portion would be the proteins that we find embedded in the lipids themselves. So there's one type of protein here. It's the transmembrane protein, meaning it's going to transcend both parts of the membrane. So it's going to be on the inner, it's going to travel all the way through into and out of the cell. We have the integral protein, which you only see in portions of the cell membrane. And then we have another one here that has a different structure to it. The glycoprotein has a channel inside of it. And sometimes we'll see that listed as a channel protein because of it. So the takeaway from here is that the cell membrane is made up of lipids and proteins. That lipid is a phospholipid, meaning there's a top portion and there's a bottom portion to it. It's almost like a sandwich. There's a top and a bottom to it. The proteins, you have one protein going all the way throughout, one protein that's just at the very top of the sandwich, and one that transcends maybe like 3/4 of the membrane. But the takeaway, once again, is that it's made up of lipids and proteins. I want to go back a second. Let me go back one more. That communication portion, you're also going to find receptors on the cell membrane. And these receptors are going to be involved in communication. So signals could be a hormone signal. These signals are going to be sent to the cell. It could be where a cell needs to take in a certain material or not, but the receptors found on the cell membrane are important in cell communication. Okay, so let's go on. We're talking about things moving in and out of the cell membrane, and that process is referred to as diffusion. There's two types of transport through the cell itself. There's passive transport and there's active transport. In passive transport, no energy is needed. So energy is not required for this process because energy will go from an area of high to an area of low concentration. And so the molecules will naturally move from the area of high to low concentration. And so it's not necessary for energy to move materials from high to low. So diffusion is the movement of molecules from an area of high to low concentration. So we're talking molecules such as oxygen, could be a sugar compound, salt compounds, any of those could work. And then we have facilitated diffusion. Facilitated diffusion is how materials move across the cell membrane using molecules, using cell membrane. So the cell membrane is going to aid them in moving across the cell membrane. All right, let's check out this video. So we see that there are simple, let me pause this for a second. There's simple diffusion and there are facilitated diffusion. All right, and so we could see that in the simple, that's going from high to low, facilitated, you need a protein to help the molecules get across. Osmosis is when water moves from an area of high to low concentration. And so in this image, I wanted to show it to you because there are some molecules that are going to be smaller than others. And so more likely the smaller molecules are going to easily move from one area to the next. When we're talking about osmosis, we're talking about solutions. So the mixture. So in a mixture, you're going to find that some molecules are larger than others. All right, let's keep going. Let's take a look at that again. So here we are. Here we have a concentration of solution, and over, so it's high concentration. And then over here, it's low concentration. So let's watch and see. So what I wanted to point out specifically in this, like the solute is whatever is in the mixture that needed to move. So in this demonstration, we were focusing on the green molecules, the solute. But with this image, with this video, what I wanted to point out is that molecules are going to move from an area of high to low concentration. They will continue to move across the concentration gradient. They're going to continue to move until equilibrium is met. So if, let's make it more simplistic. If there were 10 molecules here, the molecules would move until you end up with 5 and 5. So molecules will move in and out of the cell membrane until it's even on both sides. And so once it's even, we said that equilibrium has been met and it's being maintained. Because if all of them, if 8 went to one side, then diffusion would start to happen in the opposite direction. So it will continue to go back and forth. So that's why molecules will move until they reach equilibrium. All right. So once again, passive transport, no energy is required for passive transport. We have osmosis, is the movement of water molecules from an area of high concentration. Whereas diffusion, as I mentioned before, just reference molecules. It could be gas molecules, sugar molecules, simple sugar compounds moving across the membrane. When we talk about osmosis, we're talking about the liquid, water, the water in the solution moving from an area of high to low concentration. So before I go on to that image, you want to think, let me show you quickly. So I have a concentration gradient right here, similar to the video we just saw. So if I have a solution here, I'm going to pretend this is my container, and let's say it's liquid and I have molecules in here. Let's put couple molecules here, but surrounding it, we'll have water molecules. My dots are going to represent my water molecules. All right, and let's put some more substance in there. And I'm going to put some substance over here also. And the small dots are representing water. If we look at this, we're talking about osmosis now, the movement of water. Side A, I'm going to call this side A, and this is side B. Which side has the most water? Think about it. My smaller dots represent the water. The side that has the most water is going to be side B, because there's less solutes in it, molecules. So I'm expecting, based on the definition of diffusion, I'm expecting the water to move in this direction from here to here. I'm expecting it to go from an area of high concentration of water into a section of low concentration of water. That's what I'm expecting based on the definition of diffusion. So we're talking about osmosis. We're looking at the water molecules in the solution. If we're talking about diffusion, if this was a question based on diffusion, you would take a look here on A. We'd be talking about those molecules, those larger molecules that's here in the solution. These molecules would then move across if I was discussing diffusion. But in osmosis, we're focusing on the liquid, on the water molecule move across. I hope that gave you just a visual and a reference on the difference between the two. So once again, osmosis, we're thinking of the movement of water molecules that's in a solution, a mixture from an area of high to low. Whereas diffusion, we're looking at the molecules themselves, like oxygen, sugar, moving from an area from high to low. This was an image that we took under the microscope. Here is an onion cell. So this is the red onion cell, and you could see the cell wall in the clear, those clear lines that shone throughout, and the red represents the cytoplasm inside each cell. So in this image, I was trying to show water moving from an area of high to low concentration. So in my first image, here's the normal cell. And so I had a high concentration of liquid inside of the cell. So I then place this onion in a salt solution. So I'm going to just use this to kind of illustrate what we did. We used the microscope and dropped some salt solution on it. So what the salt solution did was put the cells in an environment where there was low concentration of water on the outside of it. So because there weren't a lot of water in that environment I created under the microscope, the cell was then forced to move water from an area of high to low. Let's see what happened. And so this is my after picture. So if you take a look, here's my after picture. The cell wall stayed where it's supposed to stay. The cell wall does not move. Remember, it's got that cellulose, it's rigid, it's going to stay there. But you could see the cytoplasm, it's actually looking like it's shrinking on the inside. This is called plasmolysis. It's where the water, due to osmosis, is moving out of the cell. And so you could see the cytoplasm is not filling the entire section of the cell anymore. So this is an effect of osmosis where the cell was put in an environment that's referred to as a hypertonic solution, and the water was pulled out of the cell, causing the inside of the cell to shrink. And so the one I did not leave here, I'm going to try to redraw it. Let's see. So if it was put in a hypotonic situation where I have my cell, and inside my cell is my cytoplasm. Inside, if I put it in a hypotonic solution, that means I'm going to put it in an environment where there's more water outside of the cell than there is inside. So water is going to move in. And what could happen in that condition, the cell will actually lysis or go through a phase where it could actually explode. It would get so large that it would rupture the cell. And so this concept is the same reason why if you go to the hospital, you get a saline solution. If you're dehydrated, they wouldn't just inject you with regular water. You get a saline solution, right? You're given a solution that has the same concentration of water as your body cell. It's there to bring your body cells back into equilibrium, to cause molecules to move into your cell until it reaches an equal amount. If you were given like just pure, if you were just given water, regular water in your IV, your cells would rupture because of it, because you're putting the cells in a hypotonic solution, causing water to diffuse into the cell, which would cause it to rupture. Okay, so I just wanted to show you these 3 different examples of how water moves in and out of the cell. And I know in this image I use a plant cell, but the concept is the same for animal cells, like a blood cell, same thing. And so the opposite of passive transport is active transport. Now, this condition, you need energy. So this is moving molecules against the concentration gradient. So sometimes we see this in the plant root, in certain areas of your body. You'll have molecules moving from an area of low to an area of high concentration. So the way I like to look at it is, say you're riding a bike up a hill. Let's see here. Here's my hill. If you're down here on your bike and you need to get to the top, to the hill, you're going to have to exert a lot of energy to go up. So anytime the molecules have to move from an area from low to an area of high, energy is going to be required to move it in that direction. Whereas what I showed you before for diffusion, oops, I can't make it the same 2 times in a row. Need to work on my artistic skills here. Here we go. So here's high, here's low. In diffusion, you're going to pretend that you're at the top of the hill and you need to go down. You're moving molecules from an area of high to low. No energy is needed. So if you're riding a bike, you're going down the hill, you do not need to expend or use any energy to get down the hill. So it's the same thing in your body. For molecules to move from an area of high to low concentration, energy is not needed. They'll just naturally move from high to low. Whereas the opposite, if you need to move molecules from the low to the high, energy is going to be required for that. So that was a quick image just so that you could get the concept. And this happens throughout your body. It happens in single cell organism. And we also see it in plant roots and molecules moving in and out of the plant itself. Okay, so I hope that analogy helps. Speaking of it here, I have this practice question. I want you to take a look here. I'm giving you a unicellular organism. And so right away, you should be trying to identify the different portion or different parts of the cell. So here's your cell membrane. You have different structures on the inside. And so I'm showing you I have these molecules here, and I'm showing you that they're going to move out here. And B is pointing to my cell membrane or plasma membrane, as we mentioned before. So why don't I give you a second just to take in those questions. All right, so the first one, it's saying, A, we're moving materials from an area of low concentration to an area of high concentration. So anytime it's going from low to high, that's active transport. And the ways that they're different, molecules are moving from low to high. Molecule energy will be needed to move these molecules from low to high. And the second one, B, already established that it's the cell membrane. It says what specific term is best used to identify these molecules. So receptors, remember, are found on the cell membrane to help with communication in the cell. Okay, so I hope you guys got those right. And so here's another practice question to review that information. Here, I'm going to have glucose molecules, and the larger ones are starch molecules. So I'm showing you the movement. This is diffusion of materials. Diffusion, think about what I said before, high to low. Diffusion, the smaller molecules are going to more likely move from high to low. So let's see. What would it look like if I have 5 molecules of starch, 12 molecules of glucose? Glucose is the smaller out of the two. So your end product should show that the molecules move across the cell membrane. Let's put our sugar back in because it's not going to move. 1, 2, 3, 4, 5 molecules of sugar. They're going to stay there because they're too big to go across the membrane. So the things that are selected to move across are the smaller molecules. So size does influence what molecules will move across the cell membrane. So we're going to see that it will move, and we'll move to where we have 1, 2, 3, 4, 5, 6 on one side, 1, 2, 3, 4, 5, 6 on the other side. Because if you remember before, I said molecules are going to move until they reach equilibrium. So they're going to go from an area of high to an area of low concentration until equilibrium is met. Okay, so we're going to take a second to move into our next section. So I just want to double check to make sure you guys see if there's any questions before we move into our next section. Okay, and we're looking good. So let's keep going. All right, so let's go back here. Let's move all these out. Here we go. So the next section that I'm going to focus on, oops, daisy. Here we go. All right, so I just needed an extra click there. So the next section we're going to move on to is the chemical process. So just take a second to think about what we just covered in the first section, as far as all the different cell organelles, what their function are. You should be able to identify what they look like and what they do. So you could always go back and review it again. And remember, I will be having review sessions throughout the week, this Tuesday and Wednesday. So please check the calendar that you receive. And so in this next section, I want to go over some of the chemical actions. And to give you a view of where we're going with this, we just saw the organelles of the cells. So cells are made up of those organelles. Organelles are made up of macromolecules. So we're looking more on the chemical makeup right now, which are then made up of micromolecules, such as, this is where we're going to have our carbon, hydrogen, oxygen, nitrogen, phosphate, sulfur, iron, magnesium, calcium. These are all common elements for us to work with. So if we're looking at the size, we're going to have the arrow showing. We're going from the smallest component of the cell to the largest. And so we see synthesis occurring here. These molecules combine together to form the chemicals in the cell and the organelles in the cell, which make up the cell itself. All right, so let's talk a little bit of chemistry. Don't be afraid. I'm going to make it nice and easy for you. I'm going to simplify it as much as possible. So we have the compounds that we're going to focus on. We have organic compounds. So organic, we're talking about materials that are made up of carbon and hydrogen atoms. And we've been discussing sugar. So sugar was one of the examples we mentioned before. It has the carbon, hydrogen, and oxygen in it. So any compound that's made up of these elements are referred to as being organic compounds. Not that they're made organically, it's the compounds that's found in them that makes them organic. Because I do notice sometimes students will confuse the two, the difference between grown organically and something that is organic. All right, so we're going to focus on something that is organic, something that's made with carbon and hydrogen, and we'll see some oxygen compound in it also, but primarily it's that carbon and hydrogen. They have to be together. And our inorganic compounds, on the other hand, they do not contain the combination of the carbon and hydrogen together. My example that I have here is water. It has hydrogen but no carbon. Okay, and carbon dioxide is one of those that, one of those that we think of as just being organic because it has the carbon, but there's no hydrogen in it. So let's explore this a little bit more. We'll start with the inorganic. Inorganic compound, the first one is water. Water is very important in the chemical processes that takes place in our body. Water is involved in, it's the substance that molecules need and ions are needed to be transported throughout the body. And so it plays a very critical role in the chemical reaction process. And so there's two ways that this occurs. So there could be dehydration synthesis. If you take a look at the word itself, dehydration, you're thinking of being without water. So making something without water. So dehydration synthesis is a reaction in which two molecules are bound together by the removing of water. Remember, I told you that water is very important. So when you're creating a molecule, you'll see that water will get removed when the bond is being formed to form a more complex substance. We refer to that as being dehydration synthesis. The opposite of that is hydrolysis. Hydrolysis, the prefix here is hydro, referring to water. Lysis means to break down, to break something. So we're going to see in a reaction where something is broken down by adding water. So that's called hydrolysis. So we'll see maybe it's changing a starch or a starch molecule into a glucose, a simple molecule. That's called hydrolysis. Okay, that wasn't so hard. Let's keep going. Organic compounds, the most common ones that we'll cover in this session, we have carbohydrates, lipids, nucleic acids, and proteins. So our carbs, they represent a major source of energy for our activities. And so that's going to include our starches and our sugar. Starches now, there are different categories of carbohydrates. We have our monosaccharides, which looks something like this. Glucose is an example of a monosaccharide. Fructose is a monosaccharide. And so you could see the carbon chain, the backbone of that compound. So we have 6 carbon. We have the attach of the 12 hydrogens. And we have the 6 oxygens involved. So that's a monosaccharide. Next, we have our disaccharide. Disaccharides now is where we have 2 molecules. So here, by itself, we have that 1 molecule, but now we have 2 attached, hence the prefix di, representing 2 molecules of sugar. So sucrose is an example of a disaccharide. Maltose is an example of a disaccharide. Sucrose is our common table sugar. And so you could see in this image, they're showing you that the pentagon ring here, the carbon makeup of that glucose molecule. And so we have our polysaccharide. And so these are many chains of monosaccharides connected together. So you have your starch molecule here. Showing you a quick example. And remember, we spoke about cellulose. Do you remember where that was found? Right, that was found in our cell wall. That's many, many chains of monosaccharides attached together. So these are complex molecules of sugar, whereas this is a monosaccharide is a simple molecule of sugar. So remember, this will be available on Learn America for you to go back and review. So here's another way I try to simplify that image for you so that you could see it. So if we have that carbohydrate, under that category of carbohydrate could be our starch, it could have been the cellulose. To hydrolyze that means we're going to add water to break it down into smaller molecules of sugar. But if we have two small molecules of sugar, we could synthesize them by removing water to make it more complex. I hope you saw how that came together, how we had both process where water is being added and taken out, how that affects the chemical process of creating and breaking down a substance. Right? So this was a simple way of showing it to you. The process is more complex than this, but I think this gives you just a quick overview of what that process would look like. I want to add this there as to what are some good ways to recognize carbohydrates. A lot of times they end in ose. We have our glucose, cellulose, amylose, fructose, maltose, sucrose, lactose, sugar found in milk. And any of our saccharides, we have the monosaccharides, disaccharides, and glycagon, which is also found in our liver. These are all sugars. And so if you're familiar with the words, as soon as you're reading through something, if you see any of these being described, you'll know right away they're in the carbohydrate family. So it's good to create word associations. It's a good way to help you memorize materials. Let's move on now to our lipids. Lipids are referred to as our fats and oils. And fats and oils, they contain carbon and hydrogen also together. So they are organic compounds. And fats and oils also serve as a source of energy for us. And so I'm showing you that picture again, showing lipids. They are made up of smaller components of fatty acid and glycerol. So if you needed to break down the lipid into those, you're hydrolyzing it by adding water, but you can also create lipid, synthesize it by removing water and joining the fatty acid and glycerol together. I like this image. It's just straightforward, it's simple, and you could just plug in the simple compound and the complex compound. Our next category is nucleic acid. And so nucleic acid, that's our DNA and RNA. They are also found in the organic compound family. But here we're seeing some newcomers here. We have nitrogen and we have phosphorus included. Whereas before we just mentioned carbon, hydrogen, and we saw oxygen throughout. Now we're seeing nitrogen and phosphate. And so once again, in lecture 2, we'll go into more details on the DNA and RNA structure and how it's inherited. So we're going to keep on moving here. That nucleotide is made as the simple component that makes up nucleic acids. All right, so let's go to this section on proteins. Protein, we're also introducing some new elements we haven't seen before. So take a look. As you can see, the new elements, we have nitrogen and sulfur. Sometimes that's included. So protein refer to our enzymes that's made, and enzymes are used to carry out chemical reactions in the body, production of hormones, antibodies, hemoglobin, and it also helps to repair and the growth of the cell. So proteins are very important process, and they're involved in so many processes in our bodies. Let's take a quick video moment. Ready? Here we go.
The primary building blocks of proteins are the amino acids. They contain an acidic carboxyl group and a basic amino group, hence the name amino acid.
All right, so I want to pause it right here to look at the chemical structure. So here we have our carboxyl group and our amino group. Here we go. And so there are different types of amino acids. Amino acids make up protein. And so the difference, all the other components are the same, except for here, this group. That's the difference between all the amino acids. Everything else would stay the same. The carboxyl group is the same, the amino group is the same, but that R section here changes and give you the different characteristics of each amino acids. Let's continue.
Each amino acid is made of 5 components: a central carbon atom, a hydrogen atom, an amino group, a carboxyl group, and a side chain. The first carbon is a part of the carboxyl group. The second carbon to which the amino group is attached is called the central carbon. This central carbon atom is joined by covalent bonds to 4 different groups. Therefore, it is usually an asymmetric carbon. R represents the side chain, which may be a hydrogen atom or an aliphatic, aromatic, or heterocyclic compound. The primary building blocks of proteins are the amino acids.
Sorry about that. I stopped it too quick. I wanted to show you those sections.
They contain an acidic carboxyl group and a basic amino group, hence the
Just
name
take a second.
amino acid. Each amino acid is made of 5 components: a central carbon atom, a hydrogen atom, an amino group, a carboxyl group, and a side chain. The first carbon is a part of the carboxyl group. The second carbon to which the amino group is attached is called the central carbon. This central carbon atom is joined by covalent bonds to 4 different groups. Therefore, it is usually an asymmetric carbon. R represents this.
All right, so this is the section I wanted to go back to. This video was worth seeing twice because the amino acid as the basic structure of the protein, and it's an integral part of the way your body works. So it was okay with seeing this twice. That R group that I discussed before, that's different. If you take a look, that's what the video just went through, showing you that that chain, that S chain section, this particular R group, can be joined by different structures. So if you look, there's another structure here. It could be this structure here. So it's showing these are 4 examples of different types of amino acid. So the thing that differentiate one from the other is what is going to replace that R, that S chain. Good.
Side chain, which may be a hydrogen atom or an aliphatic, aromatic, or heterocyclic compound.
Okay, so proteins fall into different class and composition. So we classify them, their size, the shape of the protein, also determine its function, the composition, what it's made up of, and its function. So these are the 3 characteristics, or the 3 main things we look at when we're discussing proteins. And so protein, similar to what we just did with carbohydrates, protein fall into 2 categories. There are dipeptides and there are polypeptides. And these are the larger molecules. If you break them down, they break down into amino acids. And here's that image again that I love so much. So you could see where water, if water is added, that means it's hydrolyzed, broken down into 2 parts, or it's synthesized, where you remove water to make the protein. Let's go to enzymes. So enzymes, they help to cause reactions to occur. So let's take a look. I have another video.
Enzymes are a type of protein that increases the rate of a chemical reaction. The compounds on which enzymes act are called substrates. Each enzyme has a special region present on its surface called the active site to which the substrate binds. When substrates bind with the active site of an enzyme, it forms an enzyme-substrate complex. The binding of two substrates with an enzyme causes a stress that weakens the chemical bonds present in the substrates and enables the formation of a product. The product is then released, regenerating the enzyme for further use.
Right, so that was a quick introduction. I love this video because it's very simple and it gets to the point. You see the enzyme. The enzyme is a catalyst. It's also made of protein. And its job, that process that I was just showing you about hydrolysizing the compound or dehydration synthesis, all of that function is carried out by the enzyme. So that substrate could be the lipid being broken down into fatty acid and glycerol. Or it could have been two simple sugars could be brought together to make a starch. That all of that process is done by way of the enzyme. Let's keep watching.
Such an enzyme is called an anabolic enzyme. Similarly, when a single substrate binds with the catabolic enzyme, it results in the formation of two products. Many enzymes require the assistance of other compounds in order to become activated. Some of these compounds will firmly attach to the enzyme. Other non-protein compounds that are not firmly attached are called coenzymes. An enzyme that requires a cofactor in order to become active is called an apoenzyme. When both the non-protein and the protein parts of the enzyme are present, the whole complex is called a holoenzyme.
And so guys, these videos are available on Learn America for you to go back. I think they're very simple for you to follow. So it would be good to go back and review it. And I said I like this one because it shows the different parts of the enzyme and show the two function, whether it's an anabolic or a catabolic function.
Enzymes are...
All right. And so that connection that was just carried out is referred to as a lock and key.
The hypothesis of enzyme action was postulated by Emil Fischer in the early 1890s. According to this hypothesis, a substrate binds to an enzyme in the same way that a key fits into a lock. Each enzyme is highly specific and acts only on a single substrate. The substrate binds to a specific region of the enzyme called the active site. The active site of an enzyme is complementary to the substrate and forms an intermediate site called the enzyme-substrate complex.
That's what I was talking about before. Here's your enzyme, the light blue section. And so we saw two smaller molecules joined together. And so in doing that, we're seeing that's dehydration synthesis. And so the enzyme plays a role in bringing those two molecules together.
The enzyme-substrate complex then rearranges itself to generate the product. The enzyme is now released and is used for converting another molecule of substrate into more of the product.
And so that product was a larger molecule, so that would be your lipids, your starch, your nucleic acid, your proteins. And so this was important to show you how the enzyme actually caused that to happen. There is a specific enzyme for every substrate. If it's a different substrate, you can't use the specific enzyme for a different molecule. So, for example, the enzyme to create protein is different than the one that's used to create lipids. So it's a match. They must be specific to each other. That's why it's referred to as the lock and key method. And so the last part of the enzyme concept I want to review is the induced fit hypothesis. So we've already seen that portion. Here's the complex that's formed. And so that portion is controlled by Once again, it has to have that specific fit to make it work. All right, here's some basic enzymes that we're familiar to hearing and seeing. Amylase is used to break down starch. Maltase, for example, used to break down maltose. Phospholipid would be lipase. Proteins, protease. So these are some common ones to see. And if you notice, they all have in common ASE. ASE is a common way to see proteins, to recognize protein, but it's not the only way. Here are 3 things that will have an effect on the reaction, enzyme reaction. One is temperature. What happens is temperature, when an enzyme is active, it only works within a specific range of temperature. Once it reaches the optimum temperature, what happens is the protein that makes up the enzyme itself denatures. It starts to break down. So that connection, that lock and key connection, is no longer happening. It changes the shape of that binding site. And so the substrate is not able to bind to the enzyme. So temperature will influence the reaction, how slow, how quick the reaction will take place, or if it will happen at all. The pH is also another consideration. If something is too acidic or too basic, there's some enzymes that work only in an acidic environment, some that work only in a base environment. We see in our body, there are some enzymes that function in the stomach, that pH, the acid in your stomach, keeps those, makes the perfect environment for those specific enzymes. The amount of substrate that's present will also influence how the enzyme works. So 3 things that will influence how your enzyme will work. All right, so here's a quick question. Review site, identify what are the building blocks for starch. So if you thought about it, starch is made up of simple sugar. Identify the process that's used for building these things. You could have put, you think of anything such as dehydration synthesis, synthesis in itself, that's taken smaller molecules to make a larger one. And so the story about the canary bird, it would be using energy. State one way the cardinal used these building blocks to survive. Use starch for energy, for movement and life activities. Here's another picture I'm showing you. Here is the enzyme. You need to be able to identify that that's the enzyme substrate complex, and these are the products that's made here. And so I referred to that stomach environment and how that's affected. That affects the different enzymes that work in it. So this is a question that you could go back and refer to, and if time permits, today we could come back and check it out. Let's move on to the mitochondria. Okay, so we're going to go back and review some of the materials of the mitochondria. And before I go, let me just double check to make sure that we are all set with any questions. And we're looking okay there. So let me head back. Let me head back here. All right, so let me just head back, just clean this up a little bit so we could go into our last section for today. Okay, so we're going to go back and finish off this section by discussing cellular respiration by way of the mitochondria. And that will wrap us up for today. So hopefully you remember the few things I mentioned before about the mitochondria. And as if we have some more time, we'll head back to those review questions. If not, remember you do have a test available on Learn America to go back and practice, take a test on anything that we discussed today. And I will be available for any kind of extra help this Tuesday and Wednesday. So please check your calendar. So the mitochondria, we referenced before that it's the site of cellular respiration. And so cellular respiration is the process by which organisms use oxygen to break down glucose into molecules, chemical energy such as ATP, for cells to function and release carbon dioxide. So that was a lot of information I just put out there. So in general, we spoke in general before that the mitochondria, site of respiration, energy is produced. But I wanted to get a little bit more detail as far as now, oxygen is needed. So when you breathe in, that oxygen goes through your bloodstream, diffuses from high concentration in your blood into the cell and to the mitochondria for what's referred to as aerobic respiration. Glucose comes from the life process nutrition, where you consume, take in nutrients. Your body digests those nutrients, puts it into your blood. Your blood then takes it around through your body, through the cells, and it's diffused into the cell and enter the mitochondria. So the oxygen and the mitochondria will react together through many phases of the process in order to produce ATP. ATP is the stored molecule of energy, and the waste product of it is carbon dioxide. Carbon dioxide will then diffuse out of the mitochondria and enter into the bloodstream. The blood will then take it to our lungs, and as we exhale, we get rid of the carbon dioxide as a waste product. Let's take a look at that ATP cycle. So this ADP and ATP cycle is a process of renewing and supplying ATPs and is constantly being used in the cell. Energy input combines inorganic phosphate group to ATP to form energized ATP. So you're probably like, what is that all about? So let's break it down. We'll start here. Let's start at the bottom where we see ADP down in the bottom. So ADP is a low energy molecule that's found throughout your body. It's made up of this sugar called ribose. There go that OSE again to remind you that that's a sugar. We have adenine attached to it, and we have phosphate group. So what happens is as your food is broken down and through cellular respiration, the food is broken down with the addition of oxygen, that energy that's released, here's that energy right up here. So that energy is from the breakdown of molecules. So what that energy does, it comes into the cycle and it adds a phosphate group. Here's that phosphate group to the cycle. This is called substrate phosphorylation. And so the adding of the oxygen is referred to as oxidized phosphorylation. So once that phosphate is added, take a look. Before we only had 2 phosphate group, now we have 3 phosphate group. The ribose is the same, adenine is the same. The major difference between the two is that extra phosphate group. That extra phosphate group being added in those bonds are high energy bonds. So when you break those bonds, energy is going to be released. So energy is added from your food, from cellular respiration. Food energy is then added to ADP, turning it from a low energy form into a high energy form. That's the difference between them. And so that extra energy is carried in the bonds of that attached phosphate. ATP stands for adenosine triphosphate, the 3 phosphates, whereas ADP is adenosine diphosphate. And so what happens is when your body's ready to use that energy for cellular activity, we saw it in enzyme activity. As it's needed throughout your body, that bond is broken. And so as the the high energy bond is broken, energy gets released. And so there go that phosphate, it's broken off. So once the phosphate is removed, energy is released, bringing it back to ADP. And so this is a constant cycle in your body. It's ADP, energy is introduced, taking it to ATP, high energy, your body will use the energy, bringing it back to ADP, and the process just continues over and over. So that's ATP, and it's very important because that's the energy molecule. And so here I created this chart to kind of give you an overview of cellular respiration in the cell. And you could see, here's the food I was talking about, glucose, diffusing into the cell. Once glucose get into the cell, it's converted to pyruvic acid. And so maybe 2 molecules of ATP is made through that process of converting the glucose into pyruvic acid. That process of converting glucose into pyruvic acid is referred to as glycolysis. I lost my S here. So that's glycolysis. This process takes place in the cytoplasm, not yet in the mitochondria. And so if there's a low oxygen environment or an organism that relies on low energy, in our cell, we have a process that's referred to as anaerobic respiration. So we're going to follow that up. And so this is where there's low energy. Low or no oxygen present. This process will then cause fermentation to occur. There's 2 types of fermentation. There's alcohol fermentation. And so that's when, let's see, alcohol is produced. And so we'll see people like if you're someone that's into creating like wine, this is a process that you would use. You would take the grapes through for a fermentating process. And so you would want to keep the oxygen low or none at all. And so yeast is probably added. So the yeast will go through, use the sugar from the grapes to produce the alcohol. It's one example. In our body, you ever feel that pain in your muscle? If you're doing a strenuous activity, it's referred to as muscle fatigue. That's lactic acid building up in the muscle. And that's due to anaerobic respiration. And so we see this in muscle fatigue that we experience, but also you could use this to make products such as cheese and yogurt, lactic acid fermentation is used. So that's as a result of anaerobic respiration. The one we focused on today was aerobic respiration, where oxygen comes into play. And so that oxygen then enters into, here's my mitochondria down here. And so the oxygen enters in, that pyruvic acid converts into acetyl-CoA. Acetyl-CoA is now a different form of that original sugar that entered the cell. And so the acetyl-CoA would then transform and turn into different forms based on the Krebs cycle and the electron change cycle, which are 2 detailed cycles in themselves. The waste product from that is where you'll have carbon dioxide being released. We spoke about that. You also get carbon dioxide being released from anaerobic respiration. But the major difference between anaerobic and aerobic, it's where it's occurring and also the amount of energy that's made. 36 molecules of ATP. So you get more molecules of ATP through aerobic respiration than you do in anaerobic respiration. So it's a lot, but this was a way I tried to simplify it so you could see the flow chart. So here's an example question of anaerobic, that it occurs in the mitochondria. So if we were to say what products would come out, you'd have your carbon dioxide, water, and your ATP molecules would come out. All right, let's see if we have time for a video. Yes, let's go for it.
Cellular respiration is a catabolic process in which food is broken down to produce energy in the form of ATP molecules. In plants, this energy is used in the process of photosynthesis as well as in the breakdown of sugars. Cellular respiration takes place in 3 different steps: glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis takes place in the cytosol of the cell and does not require oxygen. Here, glucose is broken down into molecules of pyruvate. It is done by the phosphorylation of the 6-carbon glucose molecule with the help of a phosphate group from an ATP molecule. This makes glucose chemically active, converting it into an isomeric form of fructose. Through a series of enzymatic reactions, the fructose is broken down into 2 molecules of pyruvate. After several enzymatic processes, pyruvate is ready for the next stage of reactions. During the process of glycolysis, there is a net gain of 2 ATP molecules and the release of 2 water molecules and 2 NADH molecules.
That's our hydrogen carriers.
Pyruvate enters the mitochondrion from the cytosol in order to enter the Krebs cycle and eventually the electron transport chain, which will require oxygen. Before entering the cycle, 1 molecule of carbon dioxide is removed from each of the 2 pyruvates in order to form acetyl-CoA. This is done through a series of enzymatic reactions. Acetyl-CoA goes through another series of enzymatic reactions in the Krebs cycle. 2 complete cycles produce 4 carbon dioxide molecules, 6 NADH molecules, 2 ATP molecules, and 2 FADH2
It's another
molecules.
hydrogen carrier molecule.
The final stage involves the electron transport chain, the major site for the production of energy-rich ATP molecules. This last stage is carried out by a series of electron-transferring proteins located on the inner mitochondrial membrane. Electrons are transferred and added with protons and eventually accepted by oxygen, the final electron acceptor, which then produces water. The ATP is formed by the proton motive force, which is driven by the movement of protons across the inner mitochondrial membrane. 30 to 34 ATP molecules are formed as a result of this process.
All right, and those additional 2, that the 36 I said, came from the original glycolysis process. So you can refer that. This is a great video. I like that it's simplified, the process, because they could get very detailed. So don't forget to go back to the site and review them. And so we'll move over and we'll come back to practice questions. If time permit, cells are organized in different ways. So we have cells, they'll come together. I'm sorry, before I go on to the cells, we want to make sure that you understand or just think back to all the cell organelles that we've viewed. And once again, you should be thinking, what do they look like? Can I identify the different cell structures? Do I know what they do? How do they function? Do I understand the chemical reaction process as we're moving forward? And don't forget to take those practice tests from the site this week and meet me for some practice time. So we see that all of those organelles work together to make up cell, and the cells will come together to form tissue. Tissues come together to form organs, organs together to form organ systems. The organ system makes up the organism. So we see there are different types of tissues. There's connective tissues, and so this is the tissue that's found in your bone, cartilage, blood structures. The function of your connective tissue is for binding and support, protect and transport, and also for insulation. Another type of tissue we see is muscle tissue. And muscle tissue is there to support the body for movement. We have different types of muscles. There is the voluntary muscle, which is also referred to as a striated muscle. There's smooth muscle, which is involuntary. These are the ones we don't think about using, whereas a striated, we find attached to bones in our body. Those are the ones we have to learn to use. But the smooth muscles, it's involuntary. We don't think about them. Found in our blood vessels, our diaphragm. Those are examples of our smooth muscles. They're found in our organs. Cardiac muscle, that's our heart muscle. And that's also an involuntary muscle. And so we can see that the muscle cells, they're going to form muscle tissues, which will make up the organs such as the heart. The heart then makes up part of the circulatory system, and the circulatory system is one of the many systems in the body. So fascinating. We have nervous tissue. Nervous tissue, they make up the nervous system, which we will, when we get to the body system, get into more detail. And so their job is really to send electrical impulses throughout the body. We have what you call it, animal tissues. Animal tissue, they're different kinds. So I just want you to take a quick look at some of these.
An animal body consists of 4 types of tissues: epithelial, connective, muscular, and nervous tissue. Both internal and external structures of the body are surrounded by epithelial tissue. Protection and absorption are some of the functions of these tissues. Connective tissues bind organs together, form blood, and store fats. Muscle contraction and relaxation occur with the help of muscle tissues. The skeletal and cardiac muscle tissues have different characteristics, but perform similar functions in bones and in the heart. While the nervous tissues facilitate conduction and the integration of stimuli, they also protect and support nerve cells.
Okay, so that gave you an image, a visual of those different animal tissues that we spoke about. And it's important that we build up to tissues in this session, because the cells, very detailed, or we went over in details the organelles that make them up and how they work together to maintain balance and homeostasis in the cell. And now we're taking it to that next level where now we're seeing that the cells are going to work together to form these different tissues in the body, which eventually will take us to organs and us exploring the different body systems. So I wanted to take you to that spot. So remember, we have a couple minutes left before we wrap up this session. So I wanted to just show you a couple of things that as you're reviewing this week, that you'll go back and look at them. And as I said before, guys, remember this is the first 2-hour session, and we have several more to come. We have 3 more 2-hour sessions. So you want to check those calendars out. You want to take advantage of the 1-hour review sessions. So after every lecture, for example, for the next 2 weeks, I'll be giving out review sessions. We have 2 this week, Tuesday and Wednesday, and then the following week, we have another 2 hours. So check those calendars, and don't forget to do the practice questions. And so here's an example of maybe a practice questions you'll come across. Here's one on cellular respiration, where I take you through. Here we're showing you that energy plus glucose. Here's that enzyme that we focused on. All right, that enzyme changes the glucose, giving off energy. It changes that glucose into pyruvic acid. From the video, we saw that enzyme come into play again. But at the end here, I have lactic acid and alcohol. So by seeing those right away, you should be thinking, is this aerobic respiration or anaerobic respiration? The clues should be that you see lactic acid, you see alcohol right away. You should know that's anaerobic respiration. Oxygen is not present. So even if I propose you come across this question as you're studying, taking your test, you'll see in the skeletal muscle tissue, if there's insufficient oxygen, what would happen to cause muscle fatigue? So this is another way you may see the review questions presented. And so what will go in that section would be lactic acid. All right, I showed you different ones. So here's another type of question you may come across. You may come across where you see the different enzymes we spoke about, where they're located. Now, this question is focusing on, by looking at this, you could see it's referring to the different things that can influence or affect the enzyme action. So if you read through this, I have a little scenario description, and then now there's specific questions to go through and answer. How would these different environments influence how these enzymes would work? So we're connecting the concept of what the enzyme is and how the environment can influence them. So just quick examples. As I said, I would like for you guys to go out and do those practice questions so we could regroup and go over them. So I believe we've come to the end of our first sessions. Remember, if you have any questions, you could send them to me at the email. So you want to double check, again, your checklists that you receive. And I hope to see you later and for our next session. Good luck with everything. Bye-bye.